Okay. Hi, everyone. Welcome to the HEAS Seminar in Ancient Genomics, where we welcome Irene Gallego Romero, who is the head of the Human Genomics and Evolution in the St. Vincent Medical Research Centre in Australia, where she has been working for quite a while at the University of Melbourne. She also has an affiliation at the University of Tartu. And so she's working in the evolution of gene expression, also in relationship to introgression, which I think is a very exciting topic. And so we're looking forward to your talk.
Thanks. Thanks, Martin. Thanks for the invitation. Also, I'm really happy to be here. So I put together perhaps too many slides, but we'll do our best. I might skip through some bits and pieces. But thanks again if you're coming down. There we go. Same. Okay, so I'm going to be speaking by saying some pretty obvious things to make sure we're all on the same footing, but human genomic data sets really don't capture the existing breadth of human genetic diversity. So this is a slide, some of you might have seen this image from, this is from a GWAS context. So the provenance of GWAS samples is primarily, the provenance of samples used in GWAS is primarily European, individuals of European genetic ancestries, which is not representative at all of human population. So Europeans make up around 16% of the human population, or people of European descent make up around 16%, but they also make up around 90, 85, depending on the year you count, percent of samples used in GWASs. So our understanding of biology of complex traits, of the genetic architecture of complex traits, is really biased towards Europeans of individuals of European ancestry. Asia is either here in blue, or green, depending on whether you want East Asia or South Asia. Southeast Asia, which is both neither of those, doesn't even count in most of these figures. And so it's really underrepresented. This is true at both the DNA level and the RNA level. So this is data. This is the predicted genetic ancestry of all the RNA-seq samples available in at the SRA, in the public genomic database. This is from work from Casper Hansen's group over at Johns Hopkins. And again, you can see that most RNA-seq samples are again biased either towards admixed American individuals who will be of either European, African American, and perhaps a little bit of Native American ancestry, or just, or of straight up European ancestry. And again, we're really not thinking about Asia in general, and much less Southeast Asia. A couple of years ago, me and a collaborator decided to look at the metadata for each of these samples. And very, very few of them have population descriptors. So if you wanted to go public, like reuse publicly available data and grab data from SRA, being like, I want every individual of East Asian ancestry in SRA, you'd be able to find around 1,000 samples that are labeled where like provenance of samples is really poorly recorded in SRA, RNA-seq. And again, the parts of the world that I work in, Southeast Asia, there is around 70 samples from Southeast Asia in all of the SRA, where there is about 750,000 RNA-seq samples. And so I keep saying, oh, wow, okay, Southeast Asia is missing. Why is Southeast Asia interesting in the first place? I think is a question that is perfectly fair to ask and perfectly worth asking. And so let me tackle that, right? So island Southeast Asia, which is the part I'll focus on, is made up out of Indonesia and the Philippines, Papua New Guinea, Timor-Leste, Brunei, and a couple of other small states. It's home to around 500 million people. That's around 7% of the global population. And Indonesia itself, which is a country where I do a lot of the work like most of my work actually, is the world's fourth largest country by population. It's home to around 280 million people, 17,000 islands, 700 languages, so huge genetic, cultural diversity. But again, no representation at all. And being based in Australia, for us, there's also an argument here that around 2% of individuals living in Australia, of Australian citizens, were born in either the Philippines, Indonesia, or Papua New Guinea. So as Australia tries to incorporate genomic medicine into its sort of public healthcare system, there is a huge argument for making sure that these people are also represented in the databases that are being used to build these resources, right? Indonesia, also, in case you don't remember what it looks like, which who could blame you, is a huge country. So here it is to scale relative to Europe. Jakarta is lined up over sort of London over here, kind of. And so again, it's a very, very big country. We're going to talk about a place in here. So keep in mind the distances that we're talking about. We're going from London to somewhere far, almost basically actually at the southern tips here of Russia. So it's huge, right? And then from a population genetic standpoint, this part of the world is super interesting. So If you look at this figure and you focus only on the black arrow, no other arrows, please. This sort of tracks the first migration of people into this part of the world around 60,000 years ago, give or take, where we had these migrants coming out of Africa, very successfully expanding all the way and into sort of New Guinea Island here, so into Sahul, right? So into New Guinea Island and into Australia. The sea levels were a bit different, so most of this was a single landmass, but there were still significant challenges, like you still had to cross significant water barriers and things like that. Somewhere in here, I don't know if it happens here, or if it happens to the left over there somewhere, or if it happens, confused, no, there we go. If it happens here and also here and there or whatever, there is gene flow from Denisovans somewhere. Don't know when, but what we know is that, of course, it does happen, right? Much, much more recently, between around 10,000 to 5,000 years ago, you get multiple waves of migrants. The biggest one is the pink one here coming from, these are farmers, they're Austronesian farmers that are coming from what today we would call Taiwan into primarily the Philippines, and then very successfully settling the western half of the Indonesian archipelago and sort of remote Oceania over here somewhere, but sort of missing New Guinea Island, which is this one here with the Papuans label, and not really settling eastern Indonesian archipelago. You also get a couple of other farmer migrations. So these over here, these red and green arrows, but primarily what all of this population movement gives rise to is this beautiful geographically structured genetic admixture line, where the western bits of the Indonesian archipelago people there are primarily from that recent wave of migrants. So the pink and the green and the red. But as you transition east in the archipelago, especially as you hit sort of east of Bali over here, the percentage of Papuan ancestry starts to rise, and it rises pretty steeply. And when you get to New Guinea Island and places around it, people there are of 100% Papuan genetic ancestry, right? So they're from that first black arrow. So this is, again, a very nicely structured genetic ancestry line. It's also happening in the context of a fairly homogeneous environment, at least like everything is tropical in that sense. So there is consistently high infectious disease burdens, although there is still sort of challenges between highland, lowland lifestyles, and also things like fishing. So primarily fishing lifestyles, or farmer subsistence farming, or not subsistence farming, sort of more agriculturally settled societies. And now Indonesia and also New Guinea really are in the midst of a lifestyle transition, moving towards a more westernized sedentary lifestyle. So you get this massive variation in environments, you get a massive variation in sort of small scale environment and also genetics, and you get the lifestyle transition. And despite all of that, there were no whole genome sequences from this part of the world until 2019. And there were no gene expression studies of healthy individuals from Indonesia until 2020, when we published what remains, I think, the only study of gene expression in healthy individuals from Indonesia. And it's a single set of like 117 samples from blood. So it's very much an open sort of like how genetic diversity and the environment play together in the Indonesian archipelago remains a really open question, right? And so my group tackles this question through different approaches. We do a lot of we've been working on pharmacogenetic diversity. We've been working on large-scale prediction of variants with like variant effects and things like that. But what I wanted to talk about, what I would pull together across different papers, was really focusing on the idea of Denisovan integration. What does Denisovan integration do? And so, like we said, guys, you can't see any of the titles. There, go away. So Denisovan integration worldwide peaks in Oceania. This is a figure from a review from a couple of years ago. And again, you can see that basically there is no gene flow from Denisovans into basically anybody of African ancestry or European ancestry. There's a bit of black migration, but we'll ignore that. It's very low levels of integration in all of mainland Asia, whether that be South Asia or East Asia. But even from looking at that, we know that the patterns of integration were really complex. There were multiple events in different places. And then it really, really peaks here in this part of the world, and sort of the eastern half of the Indonesian archipelago, as well as New Guinea, and then some communities also in the Philippines. And so this is the part of the world where, if we want to understand the dynamics of integration, we need to be looking. But like I told you earlier, this is a part of the world where absolutely no one is looking, because there is no data from it. And so when we think about our collective knowledge of Denisovan integration, of the consequences of Denisovan integration, there is this story from a few years ago, quite a few years ago now, arguing that some HLA alleles that are really high frequency in this part of the world are of intergressed origin. There is this other beautiful paper that came out, again, a few years ago now, showing integration into the single gene called TNF-AIP3, two coding variants that are prevalent, that are high frequency in Sahul, but also have become prevalent, have become high frequency sort of in these parts of Oceania, remote Oceania, are even seen in Maori, New Zealand individuals. And then, of course, there is, I think, the most famous example of Denisovan integration, which is this the story that Amelia Huerta Sanchez and Rasmus Nielsen published a few years ago that showed that adaptation to high altitude in Tibetans was caused by integration of an enhancer element upstream of this gene, EPAS1, and that was of Denisovan origin, and that was what allowed Tibetans to thrive at high altitude. But all three of these are single gene stories, right? Like so we haven't really got a comprehensive picture. And so a couple of years ago, Guy Jacobs, a collaborator of ours, sequenced around 50 genomes from Papuan donors. And of course, one of the things they did was look at integration. And what they plotted and what's shown in this figure is on the y-axis allele frequency. So you can see, okay, from 0 to 1, right? And then that's just a Manhattan plot showing regions of the genome that had integressed haplotypes at high allele frequencies and just plotting the gene names that go with them. And so again, we find this TNF-IP3 gene that I mentioned. So this is in New Guinea. So this is tropical integration. And there is other genes like ANCAR D36, this IST1, a few others. But like, that's it. That's all we know. And that's not very, at least I didn't find that ever very satisfying. I was like, well, that's a lot more, there is a lot of points you'll notice that don't have any gene labels near them anyhow. No, despite being high frequency. And there is also just no further follow-up on this. It's just saying these genes are near integration or introgressed haplotypes sort of overlap these genes, but we don't know which gene is the target. We don't know anything else about them. And so that's kind of one of the things that I really wanted to work on is kind of saying, okay, well, finding integration in DNA is quote unquote easy. I know it's not easy, but it's realistically compared to what comes after, it's slightly straightforward. Understanding what it does is way more annoying. Right. And so that's the part that I wanted to focus on, not so much detecting integration, but saying, okay, we found it. Now what? What does it mean to actually carry integration? Separate from all of that, my group had also been working on just identifying EQTLs, which are SNPs that predict gene expression levels of nearby genes in our Indonesian data set and comparing them with European data sets. And so what we had found was that there was significant sharing of EQTL signal between three different European cohorts. This is an Estonian cohort, this Lepik et al. GTX is European America, and Twins UK is from the UK. And so some of this is due to statistical power differences, but we nonetheless had a strong signal for there being unique gene regulatory impacts or differences in gene regulatory architecture between Indonesia and Europe. And so if you put all of these things together, you like the obvious question that comes out is, well, does integration in any way contribute to the signal? Do you get some sort of impact, functional impact of integration? Does integration act through gene regulation rather than changing protein coding genes? And so that's kind of what we're going to talk about for the rest of the time. So I'm going to tell you 2.5 stories today, focusing both on work that we've done in my lab in vitro with a collection of cell lines or a couple of other resources we put together, as well as more observational work on a single cell atlas that we've been putting, that we're about to finish, please, in a couple of weeks. But let's start with the in vitro studies using massively parallel reporter assays. So for that, I want to first of all acknowledge who did this work. So this work would not be possible at all without Chris Kinipi, who is the head of medical resources at the University of Papua New Guinea, Nicola Brucato, who is our collaborator over at Toulouse. And the experiments you'll hear about were done by Maddie Comerford and Davide Vespasiani, who were both PhD students in my group at different points in time. Yeah. So we started actually without any sort of experimental resources. We just had that set of genomes that Guy Jacobs had sequenced, so with 50 genomes from Indonesia, and we wanted to understand what they did. And to do that, because this was a few years ago, so we didn't have any sort of AI predictors or anything, and we didn't want to use any of the, or rather, most of the variants fell outside coding regions, so we couldn't do things that predict like the VEP, for instance. They weren't really applicable or usable to us. And so we turned to annotation from what's known as the Epigenome Roadmap project, which takes the genome and segments it into these 15 states. You don't have to worry about the colors, or you don't have to worry about the abbreviations or anything else. The only thing that matters is that there's different colors in this figure. And so if you think about the genome, when we think about DNA, right, here is a stretch of DNA. So what this project did was it annotated different bits of DNA. So this bit here, for instance, with this actively transcribed transcription start site. So here, somewhere over here, there is a gene that's being transcribed. This here is repressed chromatin, so all of this is not being transcribed. And then in orange here in this bit, we have something that is next to another actively transcribed region. Here is another actively transcribed region, and here is something else, some repressed chromatin. And so, okay, that's the state of the genome, but it's the state of the genome in one cell only. It's the state of the genome in a given neuron, right? So a neuron has to turn on certain genes to do its neuron things, and it wants to turn off other genes that are not essential to its neuron function. If we look at a different neuron, perhaps, then we see that some things are shared, some things are similar. So the gene on the left is doing the same thing in both neurons. It's a gene that's essential for being a neuron, whatever kind. And then things change here a little bit, but okay, not too much. Again, similar cell types. And then if we look across a big array of cell types, what we find is that the state of the genome varies significantly across different cell types. All of these cells are from the same individual. They all carry the same genome, but different cells will need to turn on different genes at different times, right? Like a kidney cell is not the same as a heart cell or as a liver cell. So what we thought we would do is say, okay, well, this is publicly available data set. It's rather more complex than it looks here, but it's the same idea. And so we thought we'd take all the SNPs that were found in Papuan individuals in that data set and simply intersect them with this as a guide to predicting what they do. And so, for instance, the SNPs all fall in the same place across the genome, across all the cell types, right? If you have an archaic introgressed SNP here, then it's introgressed in brain cells, but also in kidneys and in T cells. But only in some of these does it actually hit a region of the genome that is functional in that cell type or that is important for that cell type. And so we thought we could ask two very simple questions. First of all, we could simply ask, is there an excess or an absence of introgressed SNPs, so archaic SNPs, ASNPs for short, in state X? And so these are the 15 states that we consider. States 1 to 7 are considered to be active states. So these are states that are turned on in that cell type. And states 8 to 15 are inactive states or repressed states. So these are when that cell type is in one of those 8 to 15 states, those genes are off, right? And so we thought we could ask about that in general. And then we could get a bit fancier and say, is there an excess or an absence of archaic SNPs in state X? So for instance, in enhancers, in cell type Y, so for instance, in heart, relative to non-archaic SNPs, right? So and that would tell me if I find an excess of archaic SNPs in enhancers that are active in the heart, then that tells me that perhaps that's how, or that's the place where they may have an actual functional impact. And so we did this. Davide led this work. This was published a few years ago now. But basically, we started by answering that first question. And we did this because individuals from Papua New Guinea carry introgression from both Neanderthals and Denisovans, where we're able to use sort of each archaic legacy as its own sort of control for the other one. And so what we found was that both Denisovans and Neanderthals had a fair amount of introgression in these states here, which are active states. Again, if you ignore this last one here for a second. These three here are gene regulatory enhancers. So these are not genes themselves, but rather they are the elements of the genome that control when you turn a gene on, how much of that protein do you want to make, for how long, in response to what stimulus, whatever. So these are regulatory elements of genes. And we saw that in both, again, Denisovans and Neanderthals. We also saw a couple of other signals that are like that convince us that we were looking at the right place. Or that we were doing things correctly. But then we wanted to ask, okay, so again, enhancers, so things that control when genes get turned on, but which genes? So enhancers of what cell type? And what we found here, and these are the sort of official cell types from this data set we're working with, is that there was a lot of excess introgression in enhancers of blood and T cells. So these are immune cells, right? And this was, again, consistent across both Denisova and Neanderthal. Covered up here, sorry, by the red box, is also signaling smooth muscle from Neanderthals, but not so much from Denisovans. We don't quite know what's driving it. We also don't quite know what's driving this this mesenchymal thing here. Again, not quite clear to us, but what we did note is that Denisovans only, not Neanderthals, also had a lot of introgression into hematopoietic stem cells and B cells, which is sort of the other half of the immune system. So between the HSC and B cell signature and the blood and T cell signature, what we're seeing is a consistent signal for introgression into the enhancers of genes that are important for being a part of the immune system, basically. So immune signals. So this was very nice. And we were like, okay, that's great. It makes sense in the evolutionary story. You think like you can tell a very nice, cute story saying, oh, humans migrate and can move into a new region. It has new pathogens. It makes sense that you would inherit functional gene signals or functional anything from individuals who've been living there for a while because they're adapted to its pathogens. And so you can tell, again, you can wave your hands around and tell a nice story about evolution, and you can stop there, or you can be like, well, can we actually validate these predictions? Like, I mean, it's computational only, so can we go beyond that? And so there was a way to go beyond that some of you might be familiar with, which is this basic idea of a reporter assay, right? So when if you remember back when you were doing like molecular biology experiments in the lab as an undergrad, or like you say, I want to see if this piece of DNA, this single piece of DNA does something, you would put it into a bacterial plasmid right upstream of a reporter, which is sometimes GFP or sometimes it's luciferase, and you put that into a bacterial cell, and if the cell turns green or the cell glows, then your reporter element is active, is functional, can drive expression of this thing, right? And so this is nice, but you have to do it one at a time, and that takes a lot of time. And we had around 60, well, we had hundreds of thousands of these. If we wanted to test every single region, we'd have to test every single introgress SNP. That was a lot of SNPs to test. And so what we thought we would do was instead use what's known as a massively parallel reporter assay, which takes the same idea of the reporter assay, but as its name suggests, it's a lot bigger. And what lets it get bigger is that instead of it being a light-based assay where you have to then keep every experiment separate, what we have instead is we have a molecular barcode fused to each of the elements we want to test, so we can test a lot of elements at the same time, right? And here is in our workthrough of how it works. And so again, you take the set of sequences that you want to test for function, you attach a barcode to each of them, and you know which barcode goes with what sequence. You finish sort of building that into a vector, and like again, you keep working on that vector. You put that in cells, and so what cells you use depends on the question you want to do. So we use cells known as LCLs. These are immune cells. And then within each cell, if your sequence that you're testing here in the blue can actually drive expression of the reporter, then what will happen is that you will get an RNA transcript that includes the reporter, but more importantly, your barcode. And then you will sequence that barcode. And sequencing that barcode is like how many copies of that barcode you see is what tells you whether that sequence you're testing is actually driving gene expression or not, right? And so this is exactly what we did for testing introgress alleles at scale. So we took every single introgress allele that fell into an immune enhancer across PNG, Papua New Guinea, and had an introgress allele frequency above 0.15. So we've wanted to strike a balance between a reasonable number of things to test, but also introgress frequency high enough that we thought it wouldn't have gotten there by chance most of the time, that it wouldn't just have drifted. And then we paid a company to synthesize for us the version of the region with the introgress allele or the modern allele. And again, we fused molecular barcodes to it, right? And so the total number of sequences that we end up using, we tested 14,000 Denisovan SNPs, 11,000 Neanderthal SNPs, some controls. So in a single experiment, we're testing around 60,000 different sequences for function instead of doing one at a time. It is much more efficient than doing 60,000 of these single reporter assays in one go, right? So this is what the data looks like at the end. It might be a bit sort of obscure, but really the whole point of this figure is to show you that we can discriminate. Our positive controls have a higher activity, so they work. The sequences can actually drive expression better than our test sequences, both the random sequences, these are our negative controls, and then the two introgress sequences, the Denisovan and the Neanderthal. And so it says to us that, again, okay, the experiment works, but we want to dig deep into the sequences. The other thing, though, that you might notice is that out of the 61,000 that we tested, only around 3,600 were actually active. So most of the sequences we tested in these immune cells had no ability to drive gene expression at all, at least in this context, in this experimental context, saying to us that, okay, they're present in the genome, but just because they're near a gene doesn't actually mean, at least for in this experimental setting, that they actually impact the gene. They might have potential, but we're not seeing it, right? And so then we wanted to understand, well, okay, can we tease out some features that are associated that will let us predict whether something is active? And when we focused on, we thought about this one, is allele frequency. And so you can again make two arguments. You can think that introgressed alleles have reached high frequency either because they do something and therefore selection selects for them, or because they do nothing, and therefore they're allowed to drift to high frequency because they have no phenotypic consequences, or really the sort of the two. So either way, we thought there might be some association then with allele frequency and their likelihood of being active. But when we split the data into deciles, when we look at active to inactive, high allele frequency alleles to low allele frequency alleles, we find that it doesn't matter at all. Allele frequency did not impact, at least in our setup, the likelihood of observing function, of observing functional integration. So you're just as likely to be functional if you are segregating. Here on the leftmost spin, we have allele frequencies of 0.6, 0.7. Than if you are at 0.15, which was, I think, actually a bit surprising for us. The other thing that we found, though, was that SNPs that fall near a transcription start site, so near the beginning of a gene, are much more likely to be active than SNPs that are far away from it. And again, that's true whether you are at the Denisovan allele in purple or a Neanderthal allele in orange. And so again, that's kind of, it's more than anything else. This might seem like a really cool result, but actually, when you're in the promoter region, which is the region near the gene, tends to have bigger effect sizes, so they're easier to find than effects from enhancers that are far away. And so there is a bit of a circularity here with these elements are easier to identify, so we find more of them, but it doesn't mean that something far away isn't working, isn't functional. It just says to us that our assay is working. So looking across the entire genome, we didn't find much actually at this point in time that made us go, okay. So now what we wanted to do, though, of course, was identify differentially active SNPs. So SNPs where the Denisovan allele and the modern allele have different consequences. And so in this case, like this would look kind of like this. In your introgressed allele, you would see a lot of copies of the barcode. But for the modern allele, you would only see a few copies of the barcode. And that tells you that then not only can this allele drive function, but actually the two SNPs, not only can the SNP drive function, but the two alleles of the SNP have different impact on function, right? And so we did some testing. We used this model called MPRALM, and we found 98 SNPs that were genome-wide significant. So they're shown here. The orange ones, again, are Neanderthal. The purple ones are Denisovan. And we've labeled some of the genes. And again, what was for us reassuring was that some of these genes, especially as IFE1 and TNF-IP3, were some of our strongest signals, where the introgressed allele drove a much higher expression than anything else. But on the whole, it was not a lot of signal. Right. Nonetheless, we wanted to understand again, were there any features that were common to all of these SNPs? And so in this case, what we found actually, and I don't have to show you, what we found was that being at high frequency, especially the Denisovan alleles, were less likely to be differentially active than alleles at low frequency. So here we actually have a negative association between allele frequency and the likelihood of differential function, which again says to us that integration is tolerated so long as it doesn't do anything. If it does something, it tends to be selected against, which to us was not quite what we were expecting. The other thing that was really interesting is that now when we're looking at the transcription start site, there is very little Neanderthal signal within the transcription start site, but there is a fair amount of Denisovan signal within the transcription start site, again, suggesting different patterns of selection acting on the two different sources of integration, which we thought was pretty neat. And then one more thing that we wanted to look at was this idea of, well, how are these effects actually being realized? Like, what is the actual mechanism by which this is actually happening, right? And so we, if you think of one of the easiest mechanisms by which you can impact gene expression is by playing with transcription factor binding. Transcription factors are things that bind to DNA and then sort of set off a chain of consequences that sets off either higher expression or less expression. And so mutations in the sites that transcription factors bind can impact their like sort of gene expression downstream. So for instance, in this genome, if you mutate this T to a C, you go from high expression of this RNA to low expression of it. And in this case, again, if you mutate the A to a G, again, you lose expression, right? So you can play with expression levels of a gene simply by changing the transcription factor scale. And so we did a couple of methods. And what we ended up finding, which is very reassuring, was that in cases where the modern allele is expressed at higher levels than the archaic allele, what we found was that the modern allele was associated with a better transcription factor binding site than the archaic allele, and vice versa. When the archaic allele was associated with higher activity, then the archaic allele was also given rise to a better transcription factor binding site than the modern allele, which suggested at least a mechanism of action. And this was consistent no matter how we looked at it. There is still a bit of noise in the system, so it's not perfect ever. But you do see this general trend, which suggests that at least if integration is functional, it seems to be doing it by rewiring these expression setups. Sorry, these expression, oh my God. Sorry. these expression levels through transcription factor binding disruption, right? Which was nice. Like at least that was reassuring that we had at least a mechanism for the 98 SNPs that were actually differentially active. And so just really quickly, let me show you a couple of our examples. So for instance, our strongest hit is the SNP within an intron of this gene called IFE1, which is associated with a bunch of autoimmune disorders. And again, the Neanderthal signal across our replicates is way stronger than across the modern allele. And it's an allele that, despite being Neanderthal, it actually is very present only sort of in East Asia, Papua New Guinea, and then really, really peaks in South Africa. So it suggests that it's got some complicated history. And here's our outer allele. Here's the one that we really like, this TNF-IFP3 signal, which you can see is absolutely absent from everywhere else in the world, except for Papua New Guinea. And then you see mid-range allele frequencies in some of the HTTP populations in South Africa. And again, the strength of the signal is very different between sort of the introgressed allele and the non-introgressed allele, right? And the reason we really like TNF-IFP3 being a hit is that, like I told you at the introduction really quickly, TNF-IFP3 is known to be targeted by integration already. So we have a regulatory SNP that plays with expression levels of the gene, but in Southeast Asia or here in Oceania and even again in New Zealand, you also see these two linked coding mutations that also have been shown to impact function of the protein. The protein itself is part of an immune signaling pathway, and what this one in particular does is it makes the immune response slightly more active than it would otherwise, and so you're slightly better at dealing with infections. But the reason it was found was that actually is 4 different Maori individuals went to hospital because they had an immune overreaction. And so it's kind of like you're fine-tuning gene expression, but in the wrong context, you can push it too far and end up with an autoimmune crisis. But this was our big thing. We're like, wow, okay, TNF-IFP3 looks good. We also started playing with some machine learning predictors. So we used this algorithm called AlphaGenome, which is one of these sequence to function models where you give it a genome sequence and it spits out predictions for it. And what we found was that in both cases, both the IFE1 case and the Denisovan TNF-IFP3 case, AlphaGenome said that the SNPs that we had flagged as being most likely to be functional were the ones that had the strongest likelihood of impacting local regulatory structure. So it was a nice validation, though these predictors are still a bit rough. And then genome-wide, we took all of our sequences and threw them into AlphaGenome and asked, is there a correlation between what we see in the MPRA and what AlphaGenome predicts for function? And what we found was that in all cases, different cell types, you can see there is a very weak correlation. Like AlphaGenome is not very good yet, and MPRAs are kind of noisy, so it's expected that it wouldn't be perfect. But there is a weak correlation between the activity levels we predict on the x-axis and the activity levels AlphaGenome predicts on the y-axis. Though again, you can see that they're kind of discordant. But I think what this plot suggests to me more than, wow, they're really desperate to get this into their paper, is actually more that these methods, these machine learning methods, these computational methods, especially the generalist sequence to function methods, are really going to let us change the way in which we play and we try to understand genome function at scale, right? Like this took us about 10 minutes to run. Because if you haven't played with AlphaGenome, it's extremely well documented and super fun to use, actually. And so I think if we were to redo this in a couple of years when the models are getting better, we will start to see much better correlation. And it'll be really interesting to see the cases in which these models struggle or the cases in which the computational predictors and the experimental predictors agree or disagree and why that is. Because one thing these predictors are really bad at is response to something. So they're very good at predicting steady state, but they're really bad at predicting what happens if you, for instance, take an immune cell and expose it to a pathogen. They have never thought about this question at all, the models themselves, so they can't predict it. And so there is still a lot of ground to cover there, but I think, again, they're going to be a fun tool for us to play with more broadly, right? But I told you I wanted to do experimental work, and so we're back at computational predictors, and so we're going to set the MPRA aside for a little bit and think about, and then instead of talking more about MPRAs, I want to talk really briefly about some work we've been doing using cell lines that we've been setting up in the lab. So for the past few years, in 2018 rather, we set up a very ambitious sampling project where we would collect blood in Port Moresby, the capital of Papua New Guinea, and ship it to Melbourne and make stem cells from it. And we managed to make a panel of around 20 to 25 iPSC lines, so pluripotent stem cell lines from donors from Papua New Guinea. And so we now have cell lines that actually carry integration, that are PAP1, that are really not available. This is a genetic ancestry that's not available in any of the panels out there. So we can actually see what happens, experiment on integration in vitro, so like in its context, right? And so we've done whole genome sequencing, we've done RNA sequencing, ATAC-seq, HEAS, we're wrapping up some of these stuff. We're also doing some more sort of fancy single cell level work. And here is one of our cell lines. You can see it's a beautiful stem cell line. You've ever looked at a stem cell and it does stem cell things. But what I really wanted to show you really quickly is a small study that we did trying to work out if in LCLs, which are a kind of B cell, we could actually sort of experimentally in the lab test whether integration has an impact on gene expression levels or whether integration impacts the response to pathogens or not. And so what Maddie did was she took 9 cell lines that we had from donors at that time. So 4 of them are from highland donors, from lowland donors. One is from the Bismarck Islands here. From Bougainville. And so we stimulated them, we exposed them to 2 things. So this molecule called FSL1, which is something that should turn on toll-like receptors, which have been previously shown to be targeted by integration, and this other molecule called interferon gamma. And we asked a bunch of questions, which I don't really have time to go into today at all, including whether, like, do the LCLs respond? Yes, in general, but the FSL1, for whatever reason, didn't work. Do we see differences between the sites of PNG? Not really, no, but we have very few samples. Those ancestry impact things, not really. But the question that we did kind of feel we could say something about was whether integration itself affects the response to interferon gamma stimulation, right? Like interferon gamma is a molecule that turns on one of the most sort of commonly used viral response systems. So when your cell sees something, when your immune system sees a viral pathogen, the interferon gamma pathway gets turned on. And that's how your body responds. And so when we expose the cells to interferon gamma, we're basically telling the cells, oh, look, it's a virus, do something about it. And so to do that, we kind of had to play a little bit with working out how to assign integration in these cell lines. So when you use any sort of integration calling system or method, if you use one of the reference-free methods, it just says to you integration, you figure out where it came from. If you use the methods that use a reference genome, you have to use either the three Neanderthal genomes that are out, or the single Denisovan genome, or now there is a second Denisovan cave genome, but it's old and slightly drifted, so we haven't tried that one yet. And so you get either DNA, you can confidently label an interest segment as being either Denisovan, which is to say the segment you find in your people most closely matches the cave genome from the Denisova cave Denisovan genome, or you label it as Neanderthal, which is to say it matches the Altai and the Vindija and the Denisova genomes, the Denisova cave Neanderthal genome most closely, and that's great. But there is one problem here, which is that the Denisova cave is somewhere up here, right? Like somewhere there. And integration is happening somewhere here, or at least the samples are coming from somewhere here. And we've known for a while that the Denisova cave genome is actually a poor match for the introgressing Denisovan DNA that we find in people today. The 2 things are fairly diverged. And so we took this approach from Danat Yermakovic, who was working with Michael Daneman at the University of Tartu, where we said, okay, we have the Denisova DNA, and we have the Neanderthal calls, but Neanderthal integration, we think, mostly happens around here, which is why it's shared with everybody. And so if we have something that we're labeling as Neanderthal integration because it's more similar to the Neanderthal genomes, but it's only ever seen in Papua New Guinea, is it actually, like, what is the argument? How could it have gotten there? What is the argument for saying that Neanderthal DNA, or can you really say that, how likely is it that you'll find Neanderthal DNA here only, that is not seen anywhere else in the world, when Neanderthal integration happened either here or like maybe a bit further west? To us, it seems very unlikely. And so to us, it seems that what's more likely is that actually this PNG-specific Neanderthal integration is probably of Denisovan origin, that we're not going to label it Denisovan. We're going to think it's Denisovan, or it's more Denisovan-like, and that the failure to label it as such is driven by the fact that the cave genome, the Denisova cave genome, is not a good match for the introgressing population, right? And so having told you all of that, let me tell you that in any case, there is very little integration near genes that are implicated in the immune system in our data. So we have 9 samples, and we're simply counting how many samples are overlapped by integration. So in most cases, 0. Sometimes 1 individual carries integration, or 2 or 3 or 4. So most integration is very low frequency. And then when we look at the genes that responded, that were implicated in the response to stimulation, we found that most differentially expressed genes, again, did not overlap any haplotypes. So, okay. And so we did have some differentially expressed genes, but again, very few of them were integration associated. The ones that were include things like include well-known targets of integration, such as like OAS1, OAS2 somewhere over there, GBP1, GBP2, GBP4. This is a set of genes that are close to each other. They're physically linked, and they're all responding similarly, and a couple of other immune genes. So we said, okay, well, at least there is something here. Nonetheless, like genome-wide, statistically, there is really no selection for integration in immune genes. So you're not more likely to see integration in genes that are involved in responding to interferon gamma stimulation than not. And it doesn't really matter how you group integration, whether you take all of the integration calls or only the high confidence denisovans or only this weird PNG exclusive Neanderthal integration or proper Neanderthal integration or any combination of them. So there really is no overall enrichment of contributions from denisovans or Neanderthals into genes that are involved in the immune response. We also did struggle a bit to model this properly, though, because the problem with differential gene expression is that you build a linear model that simply says state, and so exposed or not exposed, right? And so an integration, you can add an integration term to your model. But the problem with that is that integration is not constant across the genome. And so you cannot actually add it into these linear model frameworks that test all genes at the same time, because integration state changes as you move across the genome. Like one individual may be integressed for this gene, but not the gene next to it, whereas another individual will carry one kind of integration in one gene and the other gene. And so we kind of had to be a bit more sort of low tech about it. So for genes that were differentially expressed in the response to interferon gamma, we simply do a likelihood ratio test comparing two models, one which for that gene only includes introgress state, and the other one does not contain that. And so we simply ask, when does the model perform better when we have this integration status? And so this is some of the genes that do this. And so what you're seeing here this is every single gene that I'm showing you here is there's the 4 Violin plots. This is showing gene expression after stimulation on the left and before stimulation on the right for this gene, for that gene. And what we're separating here is individuals who don't carry integration here on the left side, so in the gray, and individuals who do carry integration. And that can be sort of Neanderthal, so globally shared Neanderthal integration in yellow, like this one here, or this one here, or it can be Denisovan integration, like this one here, or it can be a mix of both. So this is, some individuals carry Denisovan integration, some of them carry that PNG unique Neanderthal integration. And so what you would like to see is, first of all, these are genes where, again, knowing the individuals carry integration helps you make a call about expression levels, right? So you can see consistently that without integration and with integration, you get different average expression levels. But what is less common is seeing an impact of integration on expression levels before and after infection, right? So for instance, in this case, these individuals have exactly the same gene expression level before and after infection, but it is a different expression level than when you don't carry integration. The same is true in other cases, whereas in some other genes, you can perhaps argue that there is an association, there is sort of an interaction between the two terms. But nonetheless, on the whole, we found this again that we have very weak evidence for differential regulation, like an impact or differential impact of integration on gene expression levels during their response to stimulation. So all in all, where this kind of left us was kind of this thinking that like our work, there is a limited role for integration in fine-tuning the immune response, that we didn't find any sort of overwhelming hits, but that there is still some potential there. That said, we are kind of limited by a small sample size, and so there might well be more signal if we had more samples, right? So really quickly then, the final bit of the talk. And so this is, like I said, this is small sample size. It's only a single cell type. It's an LCL, so it's not the best laboratory model. It's a really good laboratory model, but it's not a very good model of the immune system. It's a kind of B cell. There is many other cells in the immune system. And so what we thought we would do was build a cell atlas. And so this is the team that built this Indonesian immune cell atlas. Like I said, we're hoping to publish this and like to preprint it next month. And again, I want to highlight the 4 analysts. So one postdoc and one student in my lab, Fakhrul Lambita, and then Pai Kusuma and Monica Novita, who were based at Marin in Indonesia, as well as everybody else who contributed to the work. It's actually been, I think, logistically the hardest project I've ever done, and I'll tell you why in a second. But what we wanted to do was to build an immune cell atlas of Indonesia, spanning those 2 genetic ancestries I told you about at the beginning, the Papuan ancestry and this sort of mainland East Asian, Southeast Asian ancestry. They're sort of the old ancestry and the new ancestry. And to do that, we sample in 2 different islands in Indonesia. We have around 199 donors in total from these 2 parts in Indonesia. Within each site in Indonesia, we sample 2 sites themselves, so we can start playing sort of with these individuals will have similar genetic ancestries, but these ancestry is different to that one. And the same is true here. But within each site, within Bali or Jayapura, we have different environments, so we can start playing with gene by environment interactions, right? For all individuals, we have single cell RNA sequencing, we have genome sequencing. And again, this work was led by these 4 wonderful, very hardworking people. And it's been a culmination of like a lot of effort at this point. And before I show you the bits I want to show you for this paper, I want to just give you an idea of how hard why this was a hard project. So we started this project, I think, in 2021, and we spent maybe the first 2 years engaging with community organizations on the ground in Indonesia, building community consent for doing this kind of work, getting approval from the community elders, so like the small village council that has to agree before you contact anyone to join the group, picking sampling sites, so working in Bali, deciding we're going to work in Bali, we're going to work in Jayapura, not in a different part of Papua, etc. We also need to get permits at many different layers. So obviously with local authorities, we work with the university. All of the sampling that we do is done in collaboration with the Indonesian public healthcare system. So when we go out to the villages, they come with us, they do health checkups, whether you're donating samples or not. We'll do like a few triglyceride checks, blood pressure checks, and all of that gets included in their healthcare system. This might be the one time a year they get to see a doctor. So for us, it's important to work with the Puskesmas. Again, we consent from the communities themselves, and we need to get official approval from the communities. So it's challenging. We need to get to the site. So sometimes it's not so bad. We just drive. So in Bali, we drive. In Jayapura and Lake Sentani, we take a boat sometimes. Sometimes we'll do other things. The day of sampling, again, is more recruitment, more involved in the community. Recruitment of participants is done by the community elders themselves who are bringing the community together. And then finally, data collection itself is about 20 people like showing up there in cars, bringing and setting up this massive setup to collect not only the blood pressure itself or the blood data itself, but again, detailed sort of anthropometry questionnaires and phenotypic questionnaires. So it's a huge endeavor. And getting it done, and this is only the beginning, because what happens to us is that once we actually have the samples in Bali, this is a map of Bali, we have 2 sites, so we have access to the university in Bali. So some samples, we just have to drive down to the university and process there. But in Papua, we have no access to laboratory facilities. And so what we do is at the end of the day, someone drives down from the village, takes on a plane, takes the plane for 6 hours to Jakarta, where we're waiting to process the samples. Like it's been a logistic challenge every single step of the way. But nonetheless, it worked. So here's genotype on the individuals that we used, that we collected. They're the bigger shapes. You can see sort of our 2 Balinese sites are here overlapping each other. They have very similar local ancestry. This down here, it says RF mix output. And then here are our 2 Papuan communities. And so you can see that Sede is the one out here is more Papuan. There is a bit of integration or admixture with with East Asian ancestry here in Yoboy in the other side. But we did manage, if you've done single cell, you know I have to show a UMAP plot. And so we did manage to do single cell sequencing. We have data on around 500,000 single cell, so RNA-seq from 500,000 cells from all of these individuals, right? And so we've done a bunch of things with it. I'm going to skip this one here because I don't want to talk about it right now. But one of the things that we did was identify EQTLs that were driven, EQTLs in general. So again, EQTL, so it's the idea that you have a SNP, or knowing genotype at that SNP can help you predict gene expression levels of a nearby gene. And so we use a tool called Quasar. If you're ever doing EQTL mapping, we love this tool better than any other tool we've ever tried. But what we wanted to do was ask, well, can we find EQTLs where the SNP itself is of intergressed origin, right? And so we were able, so we do EQTL mapping, and then we do fine mapping on the EQTL. So try to find the causal SNP, and then we ask, is that SNP on an intergressed haplotype, yes or no, to simplify things. And so we find a bunch of intergressed Neanderthal SNPs. We have around 59 SNPs where the EQTL signal is really strongly associated with this intergressed signal. So we really think that difference in gene expression, for instance, in this NOM1 gene, really is driven by differences in allele frequency. Sorry, like by an intergressed allele. So those are the Neanderthal ones. Here's the Denisovan ones. You can see that, of course, as you would expect, there is no Denisovan integration into Bali. So the allele frequency for all of these SNPs is 0 in Bali. But we do have some SNPs that have hits here, that have intergressed SNPs here. And here is a different way of looking at them. We have 29. Out of this entire effort, we got 29 SNPs that are Denisovan associated. The one thing that was slightly reassuring was that they hit genes like IST1 one, which is a gene that I told you earlier is very high frequency integration in Papua. Siglec7 is another one that I quite like because a few years ago, doing bulk RNA sequencing, we had found that Siglec7 expression levels were associated with your percentage of Papuan ancestry genome-wide. So again, suggesting some sort of local ancestry contribution. And we do actually think there might be more of them. And what do I mean by that? Let me show you. So here is another gene, GBP5. Again, GBP5 is near known integration targets, GBP1, 2, and 4. And so here is our EQTL. So here on the left is the entire data set, both Balinese and Papuans together. You can see the signal is actually quite weak. There is not a strong association between genotype class and expression levels. When we look only in Bali, the association is quite strong and the signal is quite robust. But when we look in Papua, like first of all, the association is weaker, but it also seems to be going in the opposite direction. Even if you sort of say, well, you have very few cases of this genotype here, the means between the heterozygote and the alt are running in the opposite direction. And to us, that was like, what is going on here? So when we looked a bit deeper, we found that in Papua, but not in Bali, there is a second SNP that is causal, or it seems to be causal for GBP expression levels. And so here is the signal across the entire data set of this second SNP, not the first SNP. Again, strong association with expression levels. In Bali, we don't see the alternative alleles, and we only see one individual that's a heterozygote, so everybody's basically fixed for the reference allele. But in Papua, we have a bit more variation. Here are nomad allele frequencies for the SNP here, this one down here. So when I saw these, I said to Pi, like, this is Denisovan, you can stop now, like, it's fine, it's settled. Like, I'm willing to bet any amount of money you want me to bet on it, that is Denisovan. And then he did the integration calling, and it was not Denisovan. I was like, oh, okay. What happened was that, again, this is one of those cases where this allele is actually not a good match for the cave reference genome. And so it's actually coming out as, we can't really assign ancestry to it. It might be Denisovan or it might be Neanderthal. The analyses that we have don't let us assign it. So we call it putatively Denisovan. Again, looking at these allele frequencies, I think it's Denisovan, but like, I'm happy to hear opinions that disagree with me. But what's really, really nice for us, or again, the other thing that convinces me or that suggests to us that we're maybe missing some of that variation because of the mismatches of the reference genome is that this is an allele that is at really high frequency or decent frequencies in Papua. It's completely absent in Bali. And so when we look at the structure of the p-values in the locus between Bali and Papua, this is the significant hit in Bali. This is this one up here. And it's significant in both populations. But this entire set of signals here, they have really high p-values in Papua, but are just invisible in Bali. There is a big haplotype there that's driving this sort of signal that we just don't see anywhere else. And so I think as we dig deeper, or as a reference genome somehow miraculously, like the Denisovan reference genomes become more representative somehow, we'll find a few more, right? But overall, again, what I think we found was really there's a limited impact of Denisovan integration on gene expression in our healthy Papuan donors, but it's important to say that they are healthy. They're not in the middle of an immune response, and so that might change if we actually expose them to a pathogen. We did find EQTLs in previously highlighted regions through orthogonal methods. So again, it's confirming things that we've known, like validation. It's not like we're finding nothing. It seems that there's not a lot to find. Again, the lack of information on the integressing Denisovan population makes things very hard. But again, what do we expect from the resting immune system? Would this change if I took these individuals and challenged them with a pathogen? We are in the middle of doing those experiments, actually. So I'll get back to you in a little bit. But wrapping it all up, really, what we have found, I think, is that there is really no strong evidence for like a consistent, strong contribution of integration, Denisovan integration into the immune systems of Papuan individuals. But I think we do have enough examples across the different kinds of work that we've done for some like consistent small contributions to fine-tuning expression levels a little bit here, a little bit of that gene there, just no wholesale rewiring of anything. It's an argument whether actually this is, you can flip it and like we can argue about this again later, whether this represents constraint against differentially active integration, like is integration being selected again in general, or does it still have a role to play? We can, again, like I said, we can argue about this, but ultimately what I think is really that these high throughput in vitro approaches or these high like experimental approaches, cell lines or anything are going to be essential for understanding diversity in data sets that are just not like, or in populations that are just not captured by our large scale databases in genomics. And with that, I'm happy to I'll thank everybody again once more. So my team, everybody else, I'm happy to take any questions in the little time I've left.